With me is Dr. Enriqueta Felip from Bailey Institute in Barcelona, that she will be chairing the session with me. I'm Ignacio Gilbazo from Quirón Salud in Zaragoza, Spain. So with no further ado, and regarding this session about ADCs in non-small cell lung cancer, I would like to welcome to the podium Dr. Marina Garassino from the University of Chicago. She will be talking about the right place for anti-Trop2 ADCs in non-small cell lung cancer.
So thank you so much, and in particular I would like to thank Delvis, Rosario, Luis, and Steven for organizing everything, because I think that we traveled from all the continents here just for you and for the young people who are listening. So thank you so much for doing that. So here are my disclosures. And so I know that this is a repetition and a repetition and a repetition, but I think this is really important for Trop2, because you know that the antibody-drug conjugates work in two different ways. One type is with a cleavable linker, which means that the drug must be internalized to produce some activity, and then with the internalization you have all the cytotoxic activity. The other type is the type that doesn't need to be internalized and just works with the bystander effects. And when we talk about the ADCs, this is a very important definition. The second is clearly the payload. For all the Trop2, the payload is a topo 1 inhibitor, and all of them, they are IgG1, which means that basically they have a serum half-life about 3 weeks, and this is important for the toxicity. However, they differ from the DAR, and the DAR is important because the DAR is predicting the activity, but is also predicting some toxicity. So anytime that you look at an ADC, you should combine all these kind of characteristics. In non-small cell lung cancer, the only Trop2 that is approved is D2DXD, but there are also sacituzumab tirumotecan and also sacituzumab govitecan that we will see in the next presentation. Why Trop2? Because Trop2 is overexpressed in about 85–90% of the lung cancer cells. And in fact, you see that there are 3 compounds in development in lung cancer. And as you have seen, all of them are IgG1, but they differ because one has a cleavable linker and another one that is sacituzumab govitecan does not have a cleavable linker. And this is important because when we will see the biomarker, you will understand that what we can use, like the QCS and MR for one drug, can't be used for the other drug. And you see that D2DXD and SAC-T are very, very similar. So, but what they differ, as you can see, is the DAR. The other part is that I will not go through the step one by one, but you see that also the toxicity is very different across the molecules. For one, there is fatigue, diarrhea, alopecia. For D2DXD, you know that there is, if you tried, there is a lot of stomatitis, and we must be very careful also with the ILD, and it gives alopecia again. And the third, that is the SAC-TMT, that as I told you, is in a certain way similar to D2DXD. You see again neutropenia, stomatitis, and alopecia. So more or less, they have all the same, and we should be very careful with the ILD, in particular with D2DXD. Starting from the second line, that is where the story started. This is more or less are the 2 most important clinical trials. This is TROPION-Lung01 and DEVOC-01. Both of them compared the D2DXD over docetaxel. And you see that, unfortunately, both of them failed in demonstrating the benefit in terms of overall survival. Starting with the first one, this is D2DXD. This is in this trial there were patients with and without actionable genomic alterations, and patients were randomized to receive D2DXD or docetaxel. The primary endpoint, there was a dual primary endpoint that was the progression-free survival and the overall survival, and the trial showed some benefit in terms of progression-free survival, in particular in the non-squamous population. So we thought that this could have become a replacement of docetaxel, but unfortunately, the overall survival was completely negative. So this drug didn't make it to the second line in this population. But what was important is that if you look at the subgroup analysis, you see that more or less there are no signals of activity in any of these subgroups. But what is interesting is that this drug does not work in the squamous population, and apparently, as you can see also, if the number of patients are very modest, apparently there is more activity in patients with actionable genomic alteration. In fact, as you see in terms of overall survival, this drug works mainly in the non-squamous, and there is a detrimental effect in the squamous population for reasons that I don't mention also because they are partially unknown. This is the other drug, that is sacituzumab govitecan. Remember that this drug works in a different way also if there is the Trop2. This drug, again, had a positive progression-free survival, a almost positive overall survival. You see that the hazard ratio was very, very close to make it to the overall survival, but was negative. And again, for reasons that are unknown, the majority of the benefit was seen in those patients who didn't respond to the immunotherapy from the beginning. And you can see that this is visible in the non-responsive. The overall survival is better in those patients who failed the immunotherapy in very early stages. And again, if we divide those patients who had a stable disease and those patients who had a progression, the majority of the benefit is in those patients who had a progression. Also, if we found these results, the company is developing the drug again in combination with immunotherapy in the first line. Moving to the first line, again, the companies are trying to move these drugs in the first line. We have, sorry, doesn't work. You can't see the first line. So there are, again, two big categories of drug. One is with D2DXD plus or minus immunotherapy and chemotherapy, and the other one is with sacituzumab govitecan. We have very, very early signals that you can see on the right, and these results are coming from the TROPION-Lung02 trial. And you can see that there are multiple cohorts at different doses, combining with the chemotherapy and also without the chemotherapy. And you can see that in the first preliminary results, we see that the benefit of the triplet is not so high as it was expected. And so the drug development will continue just with the doublet and not the triplet, also because, as you can imagine, the toxicity is also higher. What is very interesting that is also in the first line, there was a subgroup analysis with the Trop2 NMR that we will see in a moment, suggesting that maybe we will find a biomarker that will be able to select who are really those patients who can benefit of this drug. Again, also for SAC-TMT, there are many trials that are ongoing in the space of the squamous non-small cell lung cancer, in particular in the maintenance phase, and also in the space of the metastatic non-small cell lung cancer in the population. The family of the drug that are using the SAC-TMT are called Trop2, just because so we can remember when we see the name of the trial. And when we are talking about EGFR mutations, they are called OPTI, TROP, LUNGO1, and LUNGO2, and we will see them in a moment. Moving to the EGFR space that was addressed also yesterday, we have the FDA approval for Dato-DXd in the population with EGFR mutations. This is coming from this joint analysis of the TROPION-Lung05 and the TROPION-Lung01 that, if you remember, has a specific cohort of patients who received with actionable genomic alterations. And you can see here that there is a good response in these patients with a median progression-free survival of 5.8 months and the median overall survival of 15 months. And of course, because this is chemotherapy, this is really independent by the mechanism of resistance to the EGFR tyrosine kinase inhibitors delivered in the first line. We have seen also this year results of sacituzumab tirumotecan that we call SAC-T or SAC-TMT. This is a trial that randomized patients with SAC-TMT over docetaxel in patients who progressed on prior treatment on EGFR tyrosine kinase inhibitors and chemotherapy. And you can see this is just an Asian population, so we will see how we will translate the results in the Western countries. But you see that there is a huge progression-free survival in this population, and there is also a positive overall survival. And you can see that also the response rate is 45% to 15.6%. Very interestingly, we have also the results not just of SAC-TMT over docetaxel, but we have the results also of SAC-TMT over a platinum-based chemotherapy. And primary endpoint was progression-free survival, and secondary endpoint was the overall survival. And you see again that there is quite a huge benefit of using SAC-TMT in this population. Also, if you see that the control arm didn't perform so well, and this trial is also positive in terms of overall survival. Moving to how can we select the patients considering that Trop2 is a biomarker. I will not go through all the literature suggesting that Trop2 overexpression is a very debatable biomarker, but I will go through this biomarker that I think is one of the first AI-driven biomarkers. And how is done this biomarker? We know, as I told you in the beginning, Dato-DXd to be active must be internalized in the cells. So can we measure the Trop2 also not just on the membrane, but also in the cytoplasm? So AstraZeneca developed, in collaboration also with Roche, this biomarker for which you have to stain with immunohistochemistry Trop2. After the immunohistochemistry, there is an automated system that is able to divide the tumor and the non-tumor. And once you have differentiated the tumor and the non-tumor, there is a system that is able to measure the optical density of Trop2 in the cytoplasm. As you can see, I don't know if I can show you here. And this is something that you can't see with your eyes. And this is why we need more sophisticated evaluation. And then we created like a biomarker determination for which there is a ratio between the Trop2 on the membrane and the overall Trop2 in the membrane and in the cytoplasm. And so I know that is counterintuitive, but it's positive when the ratio is less than 0.56 and is negative when the ratio is in the other direction, is less than 75% of tumor cells. There are also clearly some data. This is the retrospective analysis for which we think that this can become the biomarker. And as you can see, those patients who are Trop positive, they perform better to compare to those who are Trop negative. And what is very interesting is that if you look at the docetaxel curves, Trop2 does not predict the activity of docetaxel, suggesting that this is a sort of pure predictive biomarker and is not a prognostic biomarker. Removing the population of the non-actionable genomic alterations, the results hold, and as you can see, the Trop2 is able to select more or less the population who benefits from the population who do not benefit. I remember that when I gave this talk in WCRC, there was a huge question about if we need supercomputers or we need like data scientists to do that. Apparently not, at least for what we know. And there is also a sort of validation in the real world of the ability of the pathologists to define the amount of Trop2 positive cells compared to the machine. And apparently the concordance in these preliminary results that clearly are only 64 cases over 36 cases showed that there is a very huge concordance, and so it is possible that this is really becoming not substituting the pathologist, but this is maybe becoming a new assay for which you have just to test the Trop2 and you can integrate in the clinical practice. This is not ready for the prime time, but there are all the trials now that are using Dato-DXd that are incorporating this biomarker in the first line and in the further lines in the secondary or in the first endpoints of the clinical trials. So to conclude, the second-line Trop2 antibody-drug conjugates were positive for progression-free survival and negative for overall survival. Dato-DXd, for many reasons, is non-active in the squamous population. QCS-NMR is able to select some patients who respond to Dato-DXd, not all the patients, but some patients. And we will see that in the next clinical trial, we will see if this will remain important in the prospective evaluation. And I think that for the time being, the Trop2 ADCs are in the EGFR space, at least in the United States, where they are approved. Thank you for your attention. And I want to share some pictures of the organizers, and just to say that I am so happy to be here with you both, the godfathers. And this is why we are here, because this is really a wonderful community. Thank you for your attention.